Again, I'd like to express my thanks to everybody who's taking part in UK Biobank and all the researchers who have come here who are using it. UK Biobank was funded by the Medical Research Council and the Wellcome Trust, and it was an enormous act of faith by them. The first 10 years, which is what we are just coming to the end of, is really a time when UK Biobank is all work and no play. A lot of generation of data, but now the opportunities over the next 5 or 10 years come to really understand the determinants of a very wide range of disease. And the whole idea of UK Biobank is to create a resource that many different researchers from all around the world can use to understand the determinants of many different diseases. So this idea of an open access resource for approved research from anywhere in the world, from academic and commercial researchers, in order that we understand better how to treat and prevent disease. So we actually started here in Manchester. The first pilot study to work out how to do things and how not to do things was in 2006. And then between 2006 and 2010, with the support of the National Health Service, we wrote to millions of people around the UK who live near to the assessment centres that we set up and invited them to take part. And 500,000 did between 2006 and 2010, and many of you are here. Lots of questions, lots of measurements, lots of samples collected. And as Mike Rollins just said, with no return to individual participants taking part in terms of finding out about themselves, no feedback of any of the individual results, chiefly because we don't know what they mean. We'll find lots of things, but the whole idea of UK Biobank is to find out what those things mean. How important are differences in the blood, or, as we're now doing, differences in imaging. Consent for all kinds of different research. We're democratizing research. We're putting data, huge amounts of data, really detailed data, in the hands of lots of different researchers coming from many different fields, not just people who work in medicine, but people who work in informatics, in statistics, in mathematics, in finding patterns. And as I said, no preferential or exclusive access. It's a democratic process, and one that perhaps works better than some of our more recent examples. So we opened the resource up to researchers in the first quarter of 2012. And as Naomi Allen, who will speak after me, will tell you, it's now being used by more and more researchers in more and more parts of the world, in more and more different kinds of very imaginative ways. The beauty of this is it's not constrained by the imagination of one or two researchers or research groups. It has the imagination of the world being applied to this huge database. After having provided £60 million to set up UK Biobank from the Medical Research Council and Wellcome Trust, the funders, along with the Department of Health, found some spare money in their back pocket. And suddenly we were given money to genotype all 500,000 participants, to measure 800,000 markers across the genome. And again, later in the morning, you'll hear about how those data can be used in increasingly imaginative ways. Just to talk about the genotyping, this was a 3.5-year exercise that we're coming to the end of. And we suddenly, as I say, got this offer of the funding to genotype UK Biobank participants. Where a company, Affymetrix, turned them into data. The data then came back to the Wellcome Trust Centre for Human Genetics in Oxford, and Peter Donnelly, Jonathan Marchini, Colin Freeman, and their teams checked all those data, made sure that they were high quality, and then used the data, the measured data, to get even more information about the genotype. And we released the data on the first 150,000 participants in May of 2015, and lots of different researchers have been using it. In fact, some 300 research groups around the world have got approval to use those data. So valuable are these data in terms of research that no one wants anybody else to get it first. So we had this enormous problem at the beginning of this year of how do we get the updated data on all 500,000 people to everybody simultaneously when it's the equivalent of millions and millions of filing drawers of data. 2 petabytes of data, whatever that means, of data to all these 300 research groups have to be transferred simultaneously. So they came up with the extremely interesting idea of sending the data, but you couldn't, it was encrypted, so you couldn't actually look at it. And then once everybody had downloaded it, which was going to take about a month, which we're at the end of now, we will send the code so that everybody can unencrypt at the same time, and then it's a race to publish. Again, another lump of money just suddenly appeared, and this is the thing. Once you've got this resource, then they will come. The funders will come, many different funders will come and help to enrich it further and make it even more valuable for even more types of research. So we got funding, including from the British Heart Foundation, Diabetes UK, along with the current, the main funders, to measure some 40 or so biomarkers in the urine, in the serum, and in the red cells in the blood. These are the different kinds of markers that we're measuring. Some of them relate to cardiac disease or metabolic disease like diabetes or bone and joint or cancer or renal, so kidney or liver disease. Those data are being generated at the moment. We released the urine measurements in November of 2016. We are just near the end of this year, we're going to release what's called the HbA1c, which is like a kind of time-weighted average of your glucose levels. So it gives you an idea about diabetes or diabetes control. And then about 30 or so measures in the serum, the lipids and things like that, around the beginning of next year. A huge amount, I mean, no one's done assays on this scale. So the labs in Manchester, in Stockport, have been working away, making sure they get very, very high quality data from all of these samples. One of the things we've realized is that if we want this resource to be available for the next 30 years and people to be able to assay samples over the next 30 years, we have to use those samples very, very carefully. And we have to ensure that they're used for assays that matter. Typically, though, when you have this kind of cohort, what people say is, well, I'd really like to measure serum rhubarb in people who've got this disease and in people who haven't, and compare them, and just do it in small subsets. And the problem with that is that the assay methods can change over time, and so when you do an assay in one time and then do the same assay a few years later, the results can be quite different because the assay method has changed, not because the people or their samples have changed. And so you get a lot of bitty data. If we want this resource to be useful for many different kinds of research, with those different studies perhaps needing to being interested in the same thing that they're measuring, but to ensure that they're measured in exactly the same way with very high quality, it's much better if those assays can all be done at the same time in all the cohort, as is the case with the genotyping, as is the case with the biomarkers that we're doing currently. So rather than taking bits of the sample, allowing researchers to take a bit, use it up over time, it's better if we just wait until we can afford to do assays on the whole cohort of particular analytes. This means that we can be much more efficient. We can ensure that when we do pull a sample out of the freezer, we measure lots and lots of things at the same time, so we don't deplete it. We can improve the quality control. One of the things that we do is we pull the samples out in a random order, and then we measure the samples, the 500,000 samples, in a random order, so we know that there must be no difference in whatever we're measuring. So we can use the cohort as its own control of quality, so we can make sure that there are no systematic differences in the assays. And of course, if we go to an assay supplier and say, look, we'd like to measure 500,000 people rather than 50 people, we get a very good deal. The genotyping of UK Biobank was probably about half the cost of what it would have been normally, because we were coming in with a bulk order, if you like. But to do 500,000 assays, even on relatively inexpensive analytes, is expensive. The genotyping was over £20 million. The biomarkers, £10 million. So the question is, well, how do we do that with increasingly sophisticated assays? We talked about genotyping, but you'll hear later in the morning about measuring the exome sequence, so measuring the sequence of the gene, or whole genome sequencing, where you measure every single base pair. These are costs that are much greater. But again, this is an incredibly valuable resource, and one of the things that we're creating is a huge amount of data that's of interest not only to academic but commercial researchers. And again, you'll hear about how GSK and Regeneron are funding the exome sequencing, probably costs worth about $150 million, and those data will be in the resource for all researchers to use. And finally, one of the big changes over the last few years is funding from the Medical Research Council, the Wellcome Trust, British Heart Foundation, Department of Health, to do imaging on 100,000 of the participants. We've imaged about 15,000 participants, and in the afternoon you'll hear more about how those data are emerging, how they can be used. We've just opened the second imaging centre. The first one's in Manchester, second is in Newcastle, and that's now imaging. And the third will be in Reading. And finally, everybody is linked into their health outcome data. So death, cancer, hospitalization data, and for the first time ever in the UK, a big cohort linking into primary care data. Now, I'm not going to talk about the primary care data. Kathy Sudlow will talk about that later in the day. But we think of ourselves most of the time as one nation, but in fact, in terms of health records and the medical system, it's a devolved system. So we have to go to England, to Wales, to Scotland to get these data. We have to go to different sources to get data on cause of death, on cancers, on hospitalization, but we have that all in place, and every 6 months to a year we get updates of these data, and they go into the database, and then they're made available to researchers. So we've just had our funding renewed by the Medical Research Council, the Wellcome Trust, the British Heart Foundation, and now Cancer Research UK have become core funders of UK Biobank. It's an expensive thing to keep on the road, but the more it's used, the cheaper it becomes, if you like. So we're now looking at how we can get more information about health outcomes through linkage, then turning that health record data into really reliable information about the health outcomes, phenotyping, characterizing the health outcomes in greater detail, working out how to manage this huge database. Mark Effingham, our Chief Information Officer, has this difficult problem of how do you make data on this scale easily available? Can we keep on shipping it out to researchers, or do we have to electronically ship the researchers into the data? Finishing off these funded enhancements around genotyping, biochemical assays, and imaging over the next 5 years. And then we're always thinking about additional things. Can we monitor the heart to look at cardiac arrhythmias? Is there something in people's stool that would tell us beyond blood and urine about disease? And what are the next set of markers that we should be measuring in the blood? So biomarkers for infectious diseases. It's been said for many years that maybe infectious markers are related not just to infectious diseases, but to chronic diseases like cardiovascular disease, cancers, and many others. So would measuring those markers be helpful in studying a range of different conditions? So I'll hand over now to Naomi Allen, who is the senior epidemiologist on UK Biobank, who is going to talk about the ways in which the resource is being used, and I hope we'll have some time for some questions. Naomi.